In today's lecture, we're going to talk about basics of bioenergetics, energy transfer, and enzymes. The term that describes all chemical reactions within the cells is metabolism. Some of those reactions are spontaneous and can release energy. Others may require energy to proceed. During these reactions, complex molecules are being built or broken down. To run all those reactions, cells have to obtain energy. The major energy source for cells is glucose, since its bonds store enormous amount of potential chemical energy. Glucose is broken down during the process known as cellular respiration to release energy. The synthesis of glucose occurs during the process of photosynthesis, when energy of sunlight is used to convert inorganic carbon dioxide into glucose. Essentially, the light energy is converted into the energy of chemical bonds during the process of photosynthesis, and then the energy of chemical bonds is converted into energy of chemical bonds in the molecule of ATP during cellular respiration. ATP serves as the energy currency of a cell rather than the energy storage. If a cell has to store energy, it can store it in the form of glycogen in animals or starch in plants. Now let's take a look at the energy flow in nature. Organisms known as producers, for example, plants, produce essentially their own food, glucose, in the process of photosynthesis. As a side product, they also generate oxygen. In the process of photosynthesis, as we mentioned, the energy of light is converted into chemical energy of bonds within the molecule of glucose. Consumers, animals, consume producers or other animals and use glucose that was synthesized by producers to generate energy in the process of cellular respiration. Once both consumers and producers are dead, decomposers, such as a variety of worms and other invertebrates, fungi, bacteria, and archaea, can break down dead organisms, recycle nutrients, and release some of that energy as heat. Chemical reactions in cells are usually organized in metabolic pathways, which are essentially a series of connected biochemical reactions. The function of these metabolic pathways is to convert a molecule of a substrate or multiple molecules of substrates into products in the series of metabolic intermediates produced in each individual reaction. Metabolic pathways can be broadly divided into anabolic and catabolic. Anabolic pathways are used to synthesize complex molecules from simpler ones, and you can imagine that anabolic pathways, shown here, require energy. In other words, anabolic pathways are endergonic. Catabolic pathways are the ones in which large molecules break down into small ones. These pathways usually release energy and therefore are called exergonic. As we mentioned before, energy that is produced in catabolic pathways sometimes can be stored in complex molecules, for example, carbohydrates or fats. These molecules can be broken down, and the energy released from these molecules' bonds can then be used to produce ATP. It's important to understand that metabolic reactions are not spontaneous. They have to be catalyzed by enzymes. Essentially, every reaction in the cell is catalyzed by an enzyme. The role of enzymes is broad, and they are capable of facilitating both anabolic, endergonic, and catabolic, exergonic reactions. Now we're going to switch gears and talk about energy. The definition of energy is the ability to do work or cause the change in a system. There are different types of energy, from electrical to light, sorry for the hiccup. Different types of energy include electrical, light, heat energy. If the object is moving, it has kinetic energy. For example, a bullet, a movement of molecules, photons in electromagnetic radiation, or the water in the waterfall. The energy that refers to the potential to do work depends on the position or structure of an object. So, for example, the water behind the dam has potential energy which can be converted into kinetic, energy if the dam will be open. Another example of the conversion of potential energy into kinetic is the potential energy of chemical bonds. It can be converted into a kinetic energy of a moving vehicle when these chemical bonds are broken down during the chemical reactions in the engine of this vehicle. Chemical reactions, are characterized by the free energy or Gibbs free energy. It is used to essentially quantify the transfer of energy in chemical reactions. All transfers of energy involve losing some of it. In other words, when the energy is transferred between molecules or between objects, some of that energy is irreversibly lost as heat. Gibbs free energy takes into account the heat energy or enthalpy, H, and entropy, S. Gibbs free energy essentially is a usable energy that is available to do work in a system. In this equation that characterizes the change of Gibbs free energy during the reaction, H is enthalpy or heat, change in the heat during the chemical reaction. S is entropy, which is a measure of a disorder within the system, and T is the temperature at which this reaction occurs. So if the Gibbs free energy of reactants is greater than the Gibbs free energy of products, the energy will be released during the reaction. Since the change in free energy means the Gibbs free energy of products minus Gibbs free energy of reactants, if the energy is released during the reaction, the change in the Gibbs free energy is negative. We call this reaction exergonic. In exergonic reaction, products have less free energy than the reactants, and usually exergonic reactions are spontaneous. They do not require any energy input. If Gibbs free energy of products is greater than the Gibbs free energy of reactants, then the change of Gibbs free energy is positive, which means energy has to be invested into the reaction. In other words, if energy is used during a chemical reaction, the change of Gibbs free energy is positive, and we call such reaction endergonic. These reactions are usually not spontaneous because products have more free energy than the reactants. By and large, anabolic processes are usually endergonic, and catabolic processes are usually exergonic. Another important parameter that characterizes chemical reactions is activation energy. Every reaction needs a small amount of energy to start. It is called activation energy. So every reaction is characterized by breaking and forming a certain number of chemical bonds. To break some bonds and to form others, the molecule has to enter a transition state. To get to that transition state, the energy is required. For every reaction, the transition state has a higher energy than the reactants. In other words, activation energy, shown here, is always positive, whether the reaction is catalyzed or not catalyzed. Usually this activation energy is the energy of heat from the surrounding. Heat increases the frequency and the force of the collisions between the molecules, thereby facilitating breaking of some bonds and formation of others. Essentially, the activation energy determines how fast the reaction will proceed. Most cellular reactions are characterized by activation energy that is too high for them to run spontaneously. So in order for these reactions to occur at reasonable rates and to maintain cellular metabolism, the activation energy are lowered by catalysis. Before we proceed, we need to talk about the laws of thermodynamics. The first law of thermodynamics postulates that energy may be transferred or transformed, but cannot be created or destroyed. In other words, the total amount of energy in the universe is constant. For example, chemical energy that is stored within organic molecules, such as glucose, this ice cream here, is transformed into energy of chemical bonds within the ATP molecules, which then will be used by muscles to produce kinetic energy. Light energy is converted in the process of photosynthesis into the chemical energy of cellulose, chemical bonds in cellulose within the plants. Another important law of thermodynamics is that in every energy transfer, some amount of energy, we mentioned that before, is irreversibly lost in the unusable form, usually heat. So heat energy in this case is the energy that transfers from one system to another without doing work. During cellular metabolism, the energy is lost as heat, but it may not be necessarily bad, because that allows warm-blooded animals to maintain their body temperature. It's important to understand that the more energy a system loses to its surroundings, the less ordered and more random the system becomes. The order of randomness in the system is called entropy. The greater the entropy, the greater the disorder, and the lower the energy of the system. Since all energy transfers lose some usable energy, every energy transfer increases the entropy of the universe. So in other words, in isolated systems, entropy always increases. This is another formulation of the second law of thermodynamics. We already mentioned adenosine triphosphate several times. So adenosine triphosphate is a relatively simple molecule which phosphate bonds store enormous amount of potential energy. ATP is the primary energy currency of the cell, since it provides energy for the majority of cellular reactions. ATP consists of a nucleoside adenosine, which in turn consists of the nitrogenous base adenine, and the 5-carbon sugar ribose, is bound to 3 phosphate groups. They are named alpha, beta, and gamma, depending on their position relatively to adenosine. Alpha is the closest and gamma is the farthest. The bonds that link two phosphates, also known as phosphoanhydride bonds, contain a lot of energy. The bonds between beta and gamma and alpha and beta, so essentially this bond and this bond, store particularly large amount of energy. When the molecule of ATP is broken down, it produces adenosine diphosphate, inorganic phosphate, and releases energy. In some cases, ADP can be broken even farther into adenosine monophosphate, inorganic phosphate, and energy as well. So the products of that reaction, ADP and phosphate, have much lower Gibbs free energy than the reactants, ATP and water, and that explains an extremely high energy yield for these bonds. Molecule of ATP is very unstable, and it can spontaneously dissociate into adenosine diphosphate and inorganic phosphate. During that process, free energy is lost as heat. So ATP hydrolysis essentially fuels many cellular processes, from protein synthesis to transcription to translation. It is often called energy coupling. Energy coupling is illustrated on the bottom panel here. Solar or chemical energy is used to convert ADP, adenosine diphosphate, and inorganic phosphate into ATP. You can see that this reaction requires energy and therefore endergonic. ATP then can be used in a number of cellular reactions, for example, anabolic reactions of protein or DNA synthesis. During this coupling, ATP is hydrolyzed to ADP and phosphate, and the cycle starts again. We already covered the role of ATP, the energy definitions and transfer. Now we need to talk about enzymes. We mentioned before that cellular reactions require catalysis. Catalysts in the cell are essentially enzymes. Most of them are proteins. Some very important enzymes in the ribosome are RNA. Essentially, enzymes bind the reactants, the substrate, and facilitate the breaking and formation of bonds to make a product. It is very important to understand that they do not change the Gibbs free energy change of the reaction, the delta G. The only thing that enzymes do is they reduce the activation energy. You can see this on this graph. You see that the purple line, the higher line, is the reaction without the enzyme, while the dashed line, so this is the reaction without the enzyme, and the dashed line represents the reaction with the enzyme. Now, the process of enzymatic reaction can be schematically described as follows. The enzyme binds the substrate. Substrate binds to a location on the enzyme known as active site. That binding alters the shape of the substrate and the shape of the enzyme actually, and the change facilitates the bond breaking. That distortion of the substrate essentially reduces the activation energy, and eventually enzymes release the products. It's important to understand that enzymes do not change the ratio or the yields of the products. The only thing that they change is the rate of the reaction. They significantly speed it up. So, in some cases, enzymes can even form covalent bonds with substrate molecules, but at the end of the reaction, those bonds are broken. In other words, enzymes are not wasted or spent in the reaction and always return to the original state. They're not changed during that reaction. The specificity of the enzymes is determined by specific amino acids in the active site. Enzymes are extremely specific for the substrate that they will convert into a product. Of course, there is some flexibility that allows us humans to inhibit the enzymes, but nevertheless, enzymes are very specific. It's important to understand that enzymes have optimal conditions, such as pH or temperature. It's not just if we look at the enzyme from the human intestine, where pH is around 8, it doesn't mean that the enzyme from the intestine will not work at the pH 9 or 7. It will, but not as efficient as it will at pH 8. Same ideas about temperature. The enzymes that work best at 37 degrees Celsius will probably work at 30, but not as good. In other words, enzymes can work outside of the optimal conditions, but their performance will not be the same. Enzymes, of course, can be regulated. They can be inhibited or they can be activated. There are two types of inhibition that we're going to talk about: competitive and non-competitive. In case of competitive inhibition, the inhibitor binds to an active site, blocking the substrate from binding there instead. In allosteric inhibition, or non-competitive inhibition, the inhibitor binds to a different location. The word allosteric almost literally means different location. So inhibitor binds to so-called allosteric site. That binding changes the shape of the active site, preventing the substrate to bind. This conformational change essentially doesn't let the substrate to enter the active site. If you look at the graph here, you will see that the purple line represents the rate of the reaction how it depends on the concentration of substrate. With a competitive inhibitor, you can see that the enzyme will reach the same maximum rate of the reaction as the inhibitor, as the substrate concentration increases. However, in a case of non-competitive inhibition, the maximum rate of the reaction will be lower than for the normal enzyme because there is no competition between an inhibitor and the substrate. Another important feature of the enzymes is that they can be allosterically activated. On the panel on the left, you can see that there is an active form of the enzyme, which can be stabilized by the addition of the inhibitor. This inactive form can oscillate, periodically forming active form of the enzyme, which is stabilized by the allosteric activator. You can clearly see on that illustration that this enzyme has 4 active sites, but the activator binds not to the active site, but to some other location. These allosteric activators usually induce some kind of a conformational change, which increases the affinity, the ability to specifically bind the substrate for the enzyme. Many enzymes require cofactors or coenzymes. In this situation, the enzyme without cofactor is called apoenzyme. Once it binds cofactor or coenzyme, it becomes a holoenzyme. Apoenzyme is not functional. Only after apoenzyme binds its cofactor or coenzyme, forming holoenzyme, it becomes functional. Cofactors are inorganic ions such as iron, zinc, or magnesium, which usually stabilize the enzyme structure and help it to exert its activity. Coenzymes are organic molecules that are necessary for the catalytic activity of the enzymes. You can see that a lot of coenzymes are essentially derived from dietary vitamins, from riboflavin and thiamine to ascorbic acid and folate. The last aspect of the enzyme regulation is the feedback inhibition. The function of the enzyme can be regulated by the products of the metabolic reaction that this enzyme facilitates. This is why we call it a feedback inhibition. So, for instance, in illustration on the panel, bottom panel on the left, you can see that 3 enzymes, 1, 2, and 3, are essential to produce the end product. This end product effectively serves as the allosteric inhibitor of the enzyme 1. In other words, if the product becomes too abundant, the metabolic pathway that generates the product will be inhibited and the enzymatic activity will be reduced. The panel on the right shows an actual example of the allosteric inhibition. You can see here that the product of one of the pathways essential for the synthesis of amino acids, the pathway that produces an amino acid, isoleucine, results in the inhibition of the enzyme called threonine deaminase, which is the first enzyme in the pathway that converts threonine to isoleucine. Another example is ATP. ATP is a known allosteric regulator of some of the enzymes essential for the breakdown of glucose and generating ATP itself. When ATP is abundant, it binds to those enzymes, inhibiting their activity and therefore decreasing the rate of the product formation. Adenosine diphosphate, on the other hand, is the positive regulator, not the inhibitor, allosteric activator, for some of the enzyme that ATP inhibits. If ATP levels are high, the process of ATP synthesis will be inhibited. However, if ATP levels are low, because it's broken down to ADP, in other words, ADP levels are high, the enzymes that are necessary to produce ATP will be activated, and the cell will produce more ATP.